Hello, how's it going? So this is part 3 of a 3-part lecture series on the cell, basic cell anatomy and physiology. This lecture series is intended for my micro students in my college microbiology course, but of course anybody can watch this if they're learning about the cell, and hopefully this will be of some help and benefit to you. So we looked at prokaryotic cells in parts 1 and 2, and then in part 3 we're going to look at eukaryotic cells. So again, just to review, prokaryotic or prokaryote comes from the Greek words that mean pre-nucleus or refer to pre-nucleus. And eukaryotic or eukaryote come from the Greek words for true nucleus. So now there is a nucleus within the cell. There's a membrane-bound structure that houses the nucleic acids. And so we'll look at the eukaryotic cells and do some comparisons. On the left, we have a micrograph of a eukaryotic cell. And on the right, we have a micrograph of a prokaryotic cell, a bacterial cell. Now, in terms of the relative sizes of them, this is not to scale. The eukaryotic cell, if it were to scale the same magnification level as the prokaryotic cell, we would only see a portion of the eukaryotic cell because it's so much larger. But you notice even just from this black and white kind of blobbish micrograph from a transmission electron microscope that the eukaryotic cell is more complex. It has structures in it that you can see. There's definitely a nucleus, which is not present in the bacterial cell. So they're just right off the bat, you notice differences just by looking at these simple micrographs of both eukaryotic cells and prokaryotic side by side. Eukaryotic cells have flagella and cilia. The flagella and eukaryotic cells do not move in a corkscrew manner like they do in prokaryotic cells. They move in a wave-like manner, like a sine wave, moving up and down, up and down. And they are, in terms of their structure and the way that they are anchored into the membrane, it's quite different than they are in prokaryotic cells, but nevertheless they still carry out basically the same function. Bacteria do not have cilia. Prokaryotic cells do not, but eukaryotic cells do. Not all eukaryotic cells have cilia, but some do, just like some have flagella and some don't. The cilia are cell membrane extensions that come off the membrane of the cell, and they're used for food acquisition or moving the cell through a fluid, but not like a flagellum moves it through a fluid. They're used as, say, filtration apparatus or filtration purposes like in our respiratory tract and so on. So cilia are kind of complex structures in a way that are used for a variety of functions in eukaryotic cells. They are involved in cell transport and the movement of materials into cells, capturing things, getting them into cells. So they have a variety of functions so even though they're simplistic structures. And there are a number of diseases that are called ciliopathies in which the cilia don't form or they don't form properly. And some of these ciliopathies are lethal, they're deadly. And they can cause blindness, for example, with retinal ciliopathies where you can't see structures and you can't see because there is damage or the cilia are not being produced in the retina that enable us to see. Flagella and cilia have structural components called microtubules, especially in flagella. They're made of the protein tubulin, and with the flagella and eukaryotes, they move in a wave-like manner, like I said. And they have this 9 pairs plus 2 arrangement, meaning 9 pairs of microtubules plus 2 in the middle that are called the central microtubules. They always have this structure to them, and then the protein is what forms the main body of flagella. Cilia, they have microtubules as well, but they're not as thick. They're not whip-like. They're more hair-like structures when compared to flagella. Cell wall is present in eukaryotic cells. Not all of them. Found in plants, algae, fungi, and yeast. Yeast, of course, are a group of fungi, but they're so different than many of the other fungal species. They're often taken individually as an individual group. In plants and algae, we have cellulose, and fungi, we have chitin. In yeast, we have glucan and mannan, which are protein carbohydrate compounds that are like chitin, which chitin is as well, but it's just in different proportions and different types of proteins and carbohydrates that are present. As in prokaryotic cells, eukaryotic cells have a glycocalyx, which is a sticky layer that covers the plasma membrane. From it are carbohydrates that extend from the plasma membrane that are used for cell identification, cell-to-cell adhesion, and so forth. With these carbohydrates, they're bonded to proteins and the lipids within the membrane, and they stick up off the surface of it. They're used in cell communication. They assist in what's called receptor-mediated endocytosis, bringing something into a cell. So they provide a variety of functions for the cell. And so this is a superficial layer on top of the plasma membrane where these carbohydrates are extending from it. The plasma membrane is very similar to the prokaryotic plasma membrane as well. Like I said, a cell membrane is a non-negotiable for life. And so they're going to be very similar. It's a phospholipid bilayer, just like with prokaryotes. It has proteins that are on the surface of it, embedded within it. They go the whole way through it. They have carbohydrates, like I said, are used for identification and cell-to-cell adhesion and helps with transport. And there are these complex lipids called sterols that assist in preventing lysis of the cell as well, holding the phospholipids together. Is what these sterols do. So there are a few differences between prokaryotic and eukaryotic, but the essentials, it's the same. It's a phospholipid bilayer. It's a fluid mosaic model. It's got proteins on the surface and within it as well. And it's pretty much performing the same function, enabling things to get into and out of the cell and housing all of the cytoplasm, and in the case of eukaryotic cells, all of the organelles within the cell. The plasma membrane is selectively permeable, just like with prokaryotic cells. Simple diffusion occurs, like in prokaryotic cells. There's facilitative diffusion using proteins to enable things to get in and out of the cell through these helper proteins. There's the diffusion of water, and all of that is repeating the plasma membrane function in prokaryotic cells. There is a difference now with active transport. Yes, it is active. It requires energy in the form of ATP, and yes, it uses proteins as well. But there are a few forms of active transport that are found in eukaryotic cells and not in prokaryotic cells. Endocytosis is one of them. Active transport of materials into the cell. Endo means into. So this is the process of transporting things into a cell. And endocytosis can be subdivided into two main types, phagocytosis and pinocytosis. Phagocytosis is taking in solid particles through the extension and the use of pseudopods, which means false feet. So this would be like the cell membrane extending out, engulfing a solid particle and bringing it into the cell. Pinocytosis is the membrane folds inward, bringing in fluid and dissolved substances. So when the membrane folds in, a piece of it pinches off, and then the cell membrane reforms and back together again on the outside. And then that, what we could call now a vesicle, is going to bring a dissolved substance into the cell. The cytoplasm is everything between the plasma membrane and the nucleus, and it contains the organelles, so it's all the water, it's all the organelles and dissolved compounds, everything. The cytosol is the fluid portion of the cytoplasm, so this is just the liquid, watery portion. In eukaryotic cells, there is a very intricate and important structure called the cytoskeleton, which is made of these protein filaments, really small ones, there's medium-sized one, and then there are large microtubules, which forms the skeleton of the cell, and it's the transport passage network within the cell. So things move along the cytoskeleton, and the cytoskeleton gives structure and support to the cell. A muscle cell or a muscle fiber, it has an incredible cytoskeleton within it, complex, and that cytoskeleton enables the muscle fiber to contract and relax. And so it's involved in cell contraction and movement of the membrane and so forth. So it's an extremely important and vital part of all eukaryotic cells. Sorry, I should specify. Cytoskeletons are not found in all eukaryotic cells, but especially ones that have cell walls, because they don't necessarily need it, but they do have to have it for a transport network that occurs within the cell. Within eukaryotic cells that don't have cell walls, well then yeah, the cytoskeleton is even more important there because it needs a support system within it. Cytoplasmic streaming, this is a phenomenon that only occurs in eukaryotic cells, not prokaryotic. It's a movement of cytoplasm and therefore nutrients throughout the cell. So the cytoplasm is continuously moving and transporting nutrients from one area to another, from one organelle to another, and so forth. So cytoplasmic streaming, it's like streams running through the cytoplasm. Prokaryotic cells do not contain organelles. An organelle is a membrane-bound structure within a cell. So the nucleus contains the chromosomes. The ER is a transport network, among other things. It's a very simplistic function attributed to the ER. There's smooth ER, there's rough ER, and they carry out a number of vital cellular functions. There's the Golgi Complex, which forms continuously forming membranes and secretion of substances, making these structures called vesicles. The lysosome contains digestive enzymes, which is a specialized vesicle that breaks down things within the cell. It's the cleanup crew. It gets rid of the junk, so to speak. Vacuoles bring food into the cells. They store food. They provide support. They hold water. If a plant has a poisonous substance within the cell, they're stored in the vacuoles as well. Mitochondrion, cellular respiration, and you know it as the powerhouse of the cell. So this is where the ATP is being produced within the mitochondria. Chloroplast is what carries out photosynthesis through the light-dependent reactions in the Calvin cycle. And the peroxisome, these are specialized vesicles. There are many other specialized vesicles as well. They contain enzymes that destroy hydrogen peroxide, and they oxidize fatty acids so that they do not build up within the cell. There are non-membrane bound structures, ribosomes, which is the site of protein synthesis, which we talked about with prokaryotic cells. This is where the process of translation takes place, where the RNA is read and amino acids are then brought into the ribosome. The centrosome is involved in cell division, and it consists of protein fibers called spindle fibers and these other structures called centrioles. It's an area where the spindle fibers will form and then reach out and grab the chromosomes or the chromatids and then pull them apart during the process of mitosis and meiosis. The centriole is where the mitotic spindles are formed. Centrioles are groups of microtubules that exist in pairs, and they're involved in cell division as well. So there are some non-membrane bound structures that serve a specific function within a cell that are found in eukaryotic cells as well. So here's what the nucleus looks like under the microscope. It's got a membrane around it called the nuclear envelope, and then those squiggly lines that are outside of it, those structures that are in the corners that you can see in the upper left and the upper right corner and the lower left corner, that would be the rough ER. That's a continuous membrane with the nuclear membrane as well. Within the nucleus is a dense region of RNA called the nucleolus, which is inside the nucleus. The nucleolus does not control the nucleus. The nucleus is don't want to say controlling the nucleolus, but the nucleolus is under the control, I guess you could say, of the nucleus itself. Chromatin is found within there. Chromatin is just disorganized DNA and protein. It's like a protein nucleic acid soup that is within the nucleus. Chromatin will coil up and form chromosomes during the process of mitosis and meiosis. And then there are nuclear pores that allow things to get into and out of the nucleus. There is the endoplasmic reticulum, and there are two types. There's the smooth ER, which has no ribosomes attached to its surface. The smooth ER is one of its primary functions is detoxifying substances. So a liver cell, for example, contains loads of smooth ER because that becomes the detoxification center. It's also involved in the synthesis of lipids within the cell as well. The rough ER has ribosomes all over its surface. So the rough ER is integral in the process of protein synthesis. as well. So the rough ER is involved with proteins and forming proteins and organizing proteins and contributing to the formation of them. So they both have the same name, endoplasmic reticulum, but the smooth and the rough, they carry out two quite different functions. But they're both this kind of continuous membranous structure that is, the rough ER is always found very close to the nucleus, as is the smooth ER, but not always. Ribosomes in eukaryotic cells, there are two types. In prokaryotic cells, there's only one type. There is 80S and there is 70S, and it has to do with the, what do I say, I guess you could say like the mass of these things. The 80S would be considered to be I suppose, a larger type of ribosome than the 70S. The 80S ribosomes, some of them are attached to the ER, to the rough ER, and some freely float around in the cytoplasm. Still sites of protein synthesis, that's what's going on here. In 70S, we find them in chloroplasts and mitochondria. So those organelles have their own ribosomes and can carry out their own process of protein synthesis. So these are pretty sophisticated organelles, the chloroplasts and mitochondria. The 70S is the same type as what we find in bacteria. The Golgi Complex, or Golgi apparatus, or Golgi bodies, whatever you want to call it, it's any of those. This is just a series of membranous sacs that are continuously forming new parts of itself on one side and continuously being broken down and breaking off on the other side in what are called secretory vesicles and transfer vesicles as well. So in this picture here on the left, on the side that's kind of facing the lower side, where those green, the transport vesicles from the rough ER, the rough ER is sending proteins to be processed from itself into the Golgi. The Golgi will finish the packaging and the processing of the proteins that are sent to it from the rough ER. That's one side of it. And then it will send it through the cisterns, and then it'll leave through those vesicles on the other side. So the Golgi is constantly being built up and also being broken down. So it's constantly kind of remaking itself continuously. Lysosomes contain enzymes within them that can break things down within a cell. It can also break down organelles, like you can see this lysosome is combining with this mitochondrion, and it's breaking it down, so this mitochondrion has outlived its usefulness or whatever, and so the lysosome, its membrane joins with the mitochondrial membrane, and then it spews out its enzymes and degrades the mitochondrion. They also will, whenever it's time for a cell to die, program cell death, which is called apoptosis. The lysosomes will open up and they'll release their lytic enzymes, and the cell then will die. Vacuoles are very important in plant cells and algal cells where they're holding water. So these become basically storage tank for waters and other water and other dissolved particles. In this paramecium on the right, these are food vacuoles. So they'll take in little food particles and they'll store them in these membrane-bound sacs and save them. It kind of becomes like like a stomach, I guess you could say, for the paramecium. It takes it in through this oral groove, and then it forms these food vacuoles, keeps it in there, saves the food for later, has enzymes in it that can degrade and break down those food particles that the paramecium has ingested. In plants, the vacuoles have a lot of osmotic pressure within them because they're holding water, and so this is where plants store their water. So when water is plenty, the leaves will look perky or what's called turgid. Whenever there are times of drought and or low rainfall, the leaves will wilt. And the reason why is because they're losing osmotic pressure, because their vacuoles are basically empty. And so all the cells, one by one, start to shrivel up and shrink in size, and so the whole leaf then just wilts as a result of the loss of water. And here we are with the powerhouse of the cell, the mitochondrion. This is a site of ATP synthesis. It's a large, prominent organelle within the cell, and depending on what type of cell it is, that will determine how many mitochondria are in it. So, for example, muscle cells are going to be loaded with mitochondria, skin cells not so much. So this is, it has folds within it called cristae, and in those cristae, that's where biochemical reactions are taking place in the cristae and within the matrix. Through those biochemical processes of the Krebs cycle and the electron transport chain, ATP is generated, boatloads of it are made within the mitochondrion. So the cristae are those layered folds that greatly increases the functional surface area within the mitochondrion. So it doesn't have to be enormous. It can be still very small and still be packed with these membranes that are just pumping out ATP continuously. And our final slide, the chloroplast. This is a site of photosynthesis. So there are membranes and stacks of membranes, and they pile up on top of each other. They're called thylakoid membranes. And stacks of thylakoids, they're called grana, or singular is granum. And so within the thylakoids, you have what are called the light-dependent reactions are taking place, and then in the space between the thylakoids, which is called the stroma, that's where the Calvin cycle is taking place. So this is these organelles are the sites of where plants are capable of converting light energy into chemical energy, where they're creating glucose through the process of the light-dependent reactions, producing the ATP that then is necessary for the Calvin cycle then to create glucose. And we'll talk about that process later in another chapter. So that ends our three-part lecture series on the anatomy and physiology of the cell. That's a ton of information packed into one chapter. There's so much more that we could learn about, obviously, but this is just an introductory overview of the cell and the cell anatomy and the functions that occur within prokaryotic and eukaryotic cells. So I hope this was helpful to you and of some educational benefit to you. And as always, thank you for watching.